Good afternoon and welcome to the second concurrent session of the day, Neurotransmitters and Communication Between Neurons. Our presenter today is Lori Slivka from Kaiser University. Dr. Lori Slivka has earned her PhD from the University of Missouri, St. Louis. She uses neuropsychological tests and neuroimaging to examine brain integrity in individuals with health conditions that primarily impact the brain's subcortex, for example, HIV. Dr. Slivka's extensive research training greatly informs and enriches her teaching and mentoring practices. If you have any questions for the presenter during the session, please feel free to enter those into the Q&A section at the bottom of the Zoom room screen, and we will address them at the end of the talk. On that note, I will hand it over to our presenter.
All right, thank you so much for the introduction, Sarah. Can you hear me okay?
Yeah.
All right, great. Just getting up with the new technology, so wireless headphones. Just giving them a try. So thank you all for coming today. I hope you're enjoying the conference so far. We're going to do something a little bit different in this session than you may have been doing in other sessions. It's a little bit non-traditional teaching, but hopefully it's beneficial for those of you that may be teaching general psychology or introduction to behavioral neuroscience, or for those of you that are just interested in neuroscience. For a more teaching focused talk, I am actually going to be giving one in a few hours. It'll be a little bit more interactive and a little bit more about teaching. But hopefully you do learn something new from this. I study the brain. I think the brain is super fascinating. So hopefully you do too. So there we go. So Sarah provided such a nice introduction, but just a little bit about me. I did get my PhD with an emphasis in behavioral neuroscience. I'm currently a professor of psychology at Kaiser University and recently was appointed as the chair of teaching and learning at Kaiser University for the graduate school. So if you ever want to talk online teaching in general, I've been doing it for about 5 years now and love it. I know some of you, I guess, not so recently switched over to more of an online or hybrid model, but I'm always down to talk online teaching if you want. Aside from my teaching and scholarship of teaching and learning interests, my background is in neuroscience, and throughout my career, I've studied various conditions that affect a similar area of the brain. Specifically, if you flip the brain over and you look at it from underneath, it's the subcortical area. So it may seem like a wide array of conditions that I've studied, HIV, early life stress, and aging, but they all impact the similar region. So the way that I study the brain is through neuropsychological testing, so testing individuals' cognition, their thinking, and then looking at anatomical correlates. So the association between how they're thinking and what their brain actually looks like. The specific modality that I typically use is called diffusion tensor imaging. It studies the pathways that are going through the brain. So if you think of the brain like a highway system, you have your macrostructures, like your amygdala, hippocampus, and then you have this system that flows through and provides information from one area of the brain to another. So like the big structures would be like cities. I used to live in South Florida and we had a really busy highway that connected Miami, Fort Lauderdale, and West Palm Beach. And that highway system is what I study. And so if there's any like congestion, you're going to get to that location slower. So I look at essentially the congestion. I look at the integrity of the highway or the pathways that go throughout the brain. If you're interested, it's like the never ending joke, but I always tell my students if they have insomnia and they want something to go to sleep, here's my ResearchGate profile. You can just search my name. Most of my publications are under my maiden name, which is Baker. So an overview of the seminar. We see that students often are very intimidated when it comes to the neuroscience chapter in Gen Psych. And so hopefully this can, serve as a model as different like examples and ways that you may be able to teach this content without being so scary and maybe it just being a little bit more exciting. So in a prior Hawks talk, I actually talked about communication within neurons. It's in the blog if you wanted to go back and check it out. But we're going to do a quick review of what happens within an individual brain cell, an individual nerve cell. And then our focus of this talk is going to be what happens between neurons. So the communication that takes place from one nerve cell to another, essentially sending signals and sending messages for every action that we do and everything that takes place within our brain. And if there's time, last time I did this talk, I ran over a little bit, but I do want to spend some time talking about receptors and neurotransmitters. This can get kind of complex, but I want to go into some basic information that Gen Psych students or Intro to Behavioral Neuroscience students may learn, again, if we have time. So for some of you, this will be a review if you've been to any of my prior talks or if you have, specialty in this area. And then what I do when I see this specific phrase is I would ask my students, what's like the general term to describe communication within neurons? So what is the specific phrase of communication within neurons? You can just think about it in your head. So I'm going to give you a second. What am I thinking of? What is that term for communicating within an individual neuron? It's an action potential. So really what we're going to be focusing on is action potentials in individual nerve cells. So this is what a typical nerve cell or neuron may look like. They don't all look like this, but this is typically what you would see in a general textbook of the typical neuron. So I like to describe the neuron using a hand model. So if we have our hand and our arm, ignore my hairband, but the dendrites, which we're going to go into like what all of these are, but the dendrites here are my fingers, the cell body is my palm, and then the axon is my arm. So I think students find this helpful to kind of like visualize it in this way and like think about it in terms of their hand and of their arm. It kind of takes like the really complex science and makes it a little bit simpler. But in Gen Psych classes, this is often taught, these main parts of a neuron and their functions. So if we start with I'm not great with Zoom. We typically use a different platform at Kaiser. So I'm not going to point, but I'm going to guide you verbally through it. So if you look at like the middle of the palm, we see the middle of the palm and then surrounding it, we see what's called the cell body. It's the factory of the neuron. Within that is what's called a nucleus, which is that green area. It's extremely important. It contains genetic materials, including information for cell development and synthesis of proteins. Mitochondria are also housed within the cell body, producing energy and fuel. And it's surrounded by what's called a cell membrane or a neuronal membrane, which we're going to talk about a lot. It serves as a barrier that encloses the cytoplasm or the fluid within the neuron that contains the cellular material. And what it does is it excludes certain substances from getting in, and it makes sure that certain substances don't get out. It's this mechanism that's really going to start our process of our action potential when we get there, the distribution of what goes inside and outside of the cell body. I like to think of the dendrite, which is my fingers, is the listening end. I learned that, I think it was the beginning of graduate school, and I've always thought of it like that. It's the main apparatus for receiving signals from other nerve cells. This is going to make a little bit more sense when we talk about between the neuron communication. The axon, I like to think of my arm, it's the talking end. So it's a tube-like structure that carries electrical impulses from the cell body or another cell's dendrites to structures on the opposite end of the neuron, so on the opposite side of that arm, specifically called axon terminals, which then pass impulses to another neuron, which is our next step when it comes to neuronal communication. The axon is covered, most axons are covered in what's called a myelin sheath. It's a fatty material that wraps the axon. It greatly increases the speed. We see issues with the integrity of myelin sheath in conditions such as multiple sclerosis. And we can measure the integrity of this myelin sheath that helps with the speed of communication through diffusion tensor imaging. The Schwann cells are it's another type of nerve cell, and it coats or it helps form the myelin sheath. Those spaces you see in between are called nodes of Ranvier, and they help facilitate rapid conduction of nerve impulses and help with speed of communication. And then at the end of the axon, we have what's called an axon terminal, refers to the axon endings, and it helps with communication that goes from one neuron to another. So here's another typical example of a neuron. This is what it would typically look like. So although there are different classes of neurons, we can make generalizations that all neurons have three basic functions. They receive information from a neuron or a muscle, and they integrate incoming signals, determine what should be passed along and what shouldn't. They communicate signals to target cells like other neurons or muscles or glands. So in terms of communication, we start with the dendrites, typically our fingers. Information is sent through the neuron out the axon, which they're talking in. That's talking to the next cell. So a very elementary explanation of what happens throughout this process within the individual neuron is that an action potential, that communication within, occurs when a neuron sends information down the axon away from the cell body. Neuroscientists like to use the word spike or impulse for an action potential. The action potential itself is an explosion of activity that's created by what's called a depolarizing current. So the cell becomes more positive, and that's where we start. Let's start with what the cell looks like typically when it's at rest, when there's no action taking place on the individual cell. So taking us a little bit back in time, a couple slides, we're going to talk about the cell membrane. So when a neuron is not sending a signal, it's what's called, it's at rest, okay? It's defined by the charge of ions, which are inside and outside of the cell, that neuron. The inside typically is more negative relative to the outside. And this is an important point, because for a cell to fire or an action potential to take place, the cell becomes more positive. So at rest, the typical concentration, it sits at a negative value, typically between -50 and -75. So what we typically see when a cell is at rest is a concentration of sodium, potassium that create that negative concentration. So potassium are typically positively charged potassium are in higher concentration within the cell. And then sodium, which is positively charged, and some chloride are typically found higher outside of the cell. So although the concentrations of the different ions attempt to balance out both sides, they can't, because the cell membrane is like the keeper of what passes in and out. And it actively maintains this balance. So only some ions are able to pass in and some are able to pass out. So this creates a negative concentration. This is maintained by what's called a sodium-potassium pump. The sodium-potassium pump pushes sodium ions out of the cell, potassium ions into the cell, maintaining an imbalance of these chemicals. And so this happens after an action potential fires. It comes back and it helps maintain rest and ready for the next one to happen. So this is the scary figure that students often see, or my students in Gen Psych, the beginning of the behavioral neuroscience class, and they say, why am I here? I want to learn about counseling. Why do I have to do this? Well, this is how all of the action within our brain takes place. It's through what's called an action potential. So it's this figure, and this is what's happening from the time that the dendrite receives information from a neighboring cell to the time that it leaves the axon. So it takes a little bit I ended up going into this field, but I remember I took AP psychology in high school and I was always confused about this. And I try to give my students a ton of different modalities to try to learn this. So like from YouTube videos, synchronous lectures that we do, asynchronous information, the textbook, because I think once it clicks, but it's a little scary at first. You're like, why are you talking about sodium, potassium? And so keeping in mind that this is not something that everyone picks up that fast. And so they may need different modalities in order to understand this. So essentially an action potential will occur if a neuron sends information down an axon away from the cell body. This is what we see at like a microscopic level. The action potential itself is an explosion of electrical activity that's created by what's called a depolarizing current. This means some event, specifically a stimulus, causes that resting potential that cell likes to be at between -50 and -75 to get off balance, okay? So when there is a signal that comes in, that is shaken up. okay? So when the depolarization reaches negative 55 millivolts, a cell can fire what's called an action potential. It reaches what's called this threshold of excitation where it's like, okay, it's go time. We've made it past this threshold, we can fire. If it hits there, the action potential is going to fire. It's all or nothing. You're not going to get a half action potential. If it reaches the threshold of excitation, that cell is going to fire and produce signal to be sent to the next cell. So when the threshold is reached, an action potential of a fixed size will always fire. For any given neuron, the size of the action potential is always the same. There's no differing degrees. So the way that this starts is an action potential is caused when different ions cross the neuronal barrier. When that maintenance of sodium, potassium, and chloride is disrupted. So a specific stimulus to a certain point causes sodium channels to open. Because there are so many more sodium ions than outside, the inside of the neuron is negative relative to the outside. Sodium will rush in, flooding in the cell, which is what we see on this first upward slope. It's called depolarization, where the cell is becoming more and more and more positive because the sodium is coming in. So sodium has that positive charge, remember? So the neuron becomes more positive, it's depolarized. It takes a little bit longer for the potassium channels to open, but when they open, potassium rushes out of the cell. So we get to that action potential, that tip, and then we see potassium rushing out of the cell, reversing that depolarization. At that same time, sodium channels begin to close, and this causes the action potential to go back down to around negative 70 millivolts, what's called a repolarization. Oftentimes, the action potential even goes further below negative 70 millivolts, which is called a hyperpolarization. It's becoming even more negative because the potassium channels may stay open a bit long. But gradually, because of the sodium-potassium pumps, the ion concentrations go back to the resting level, and the cell gets back to around negative 70 millivolts, and it gets ready for the next action potential. But this process starts the action of what we're getting into today, which is the communication that's between neurons. This happens neuron to neuron to neuron. Action potential sends signals, leads to signals being sent to one cell, so on and so forth, till you get something that looks kind of like the Muse cover from years ago. I don't know, I used to like the band Muse. I don't know if any of you have heard of it, but they had a pretty diffusion tensor imaging picture on the front. Everyone thinks diffusion tensor imaging is really cool to look at, and it is. These colors itself don't tell us much except directionality. It can be used for surgical mapping. We really have to get in the weeds with the physics and understanding actually like the specific measurements, the specific integrity, but they sure are pretty. So many of you have seen this. Maybe it's, you know, a textbook on the wall of a lab. Maybe you do diffusion tensor imaging research, or it's the Muse album. So like I said, this picture really just tells us about directionality of the individual tracks. So these are neuron to neuron information being sent from one part of the brain to the other. Some span from frontal lobe to temporal lobe, some span from frontal lobe to occipital lobe. But they all have, the brain works together as one unit to communicate. We always, this is a side note, but going back in time to Aristotle, we thought the brain was, or our thinking, our mental capacity was located in our heart. And then we, you know, came along to understand it was in the brain. Phrenology came about, which is essentially function was located in various parts. So there's localization of function. It wasn't quite accurate, but it did lead to our understanding now that many parts of the brain do have specific functions. But where we are really now is understanding the brain works together as a unit. So while, you know, your hippocampus could look fine on an MRI, there could be disrupted connections between that and the amygdala that could be leading to some sort of symptomology. So this communication that's taking place throughout the brain is really important for us to understand. So in our brain, we have these large gray matter structures, which I mentioned at the beginning, like the amygdala, hippocampus. And there's communication that takes place between the neurons. It sends signals between these macro structures. This coordinates everything we do. Like right now, we're breathing, talking. You may be talking, maybe like, this girl's really boring. I'm talking remembering information from the past. I'm using my hands, which I always do. My chair in my department always makes fun of me and says it looks like I'm coordinating an orchestra. But all this takes place because of communication that takes place within and between neurons. Rapid speed. My brain is telling my muscles to move, my hands like this to click. It's all happening while everything else is going on at the same time, talking, breathing, my heart's beating. So I just kind of nailing it in that it's really cool. So this leads us into talking about communication that takes place between neurons. Give me 1 second. Let me adjust my screen. Oops, that's not the screen that I want to adjust. 1 second. Okay. Like a whole dual weird monitor setup going on here. So communication between neurons, also called synaptic transmission. So we're talking about two neurons and essentially marrying them. It's kind of what's happening here. So we look at the picture on the left and we see our talking end, our axon, is like kind of kissing our listening end, our dendrite. So there's really important information that's being sent from one to another through this process called synaptic transmission. So we're going to talk about this from the perspective of a typical neuron, like those pictures that I showed you. For reviewing different types of neurons, I encourage you to watch a prior talk of mine on the Hawks blog, where it was the communication between or within neurons, where I talked about that. But what I mean by typical neurons is axons sending information to dendrites. Okay, that's not always the case, but I want to think simplistically, especially if we're teaching these lower level intro classes. Sometimes it can be axon to axon, dendrite to dendrite, axon to cell body, but let's stick to the basics for now. So we discussed communication within, how this information is sent from one neuron to another to create a beautiful highway system. So in most cases, action potentials are not transmitted from one neuron to another. Information is transmitted through what's called the synapse. Typically, a chemical neurotransmitter carries information from presynaptic, we're going to get into this terminology, to the postsynaptic cell. And that neurotransmitter is really telling the next cell what to do. It's giving that information. So before we start, let's go over some definitions of terms. So the synapse, or the gap, is what we see inside that little like monster looking thing that you see on the right side. It's that space. There's multiple different ways that information can be sent from one to another. It can be sent in a chemical way or electrical. Chemical is what we think of typically. It's a neurotransmitter being transmitted. And electrical, which we're going to go into briefly today, is not as common, but crucial for survival. These designations are relative for a particular synapse. So right now I want to talk about presynaptic and postsynaptic cells. Most neurons are both presynaptic and postsynaptic and can be used interchangeably, depending on who's giving information and who's receiving information. So if we look at this picture, the cell, like the little monster that looks like it's eating the bottom monster, would be referred to as our presynaptic cell. It's taking information and sending it to the bottom little monster, which is the postsynaptic cell, receiving it. But at one point in time, that presynaptic cell you see now was the postsynaptic cell. It received information. So it gets its designation based on what it's actively doing at that time. It's not, we have presynaptic cells and we have postsynaptic cells. It just depends on the action. And then the neurotransmitters are our body's chemical messengers that are sent from whatever that presynaptic neuron is to postsynaptic dendrites. So I like to start with like basic information and then go more specific, just in thinking it's easy and then you both get on. But this gives you a snapshot of essentially what happens between neurons. So the presynaptic cell is going to synthesize and package whatever neurotransmitter it is. It could be serotonin, could be the feel-good one, dopamine, could be acetylcholine, serotonin, did I say that? GABA. But it's packaged in synaptic vesicles located in the axon terminal, which is the end part of that axon. The arrival of the axon terminal causes the release of a neurotransmitter. And then the neurotransmitter diffuses across the synaptic cleft and is received by the postsynaptic cell. Give me 1 second. I told you technology is not my friend. Just gonna grab my AirPod. Okay, so the arrival of an action potential causes the release of the neurotransmitter on the presynaptic cell. The neurotransmitter is then going to diffuse across the synaptic cleft and received by the postsynaptic cell. Then, as quickly as it happens, it reverses, in which the neurotransmitter has to be removed from the synaptic cell, so there's not continuous action taking place. We're going to go over all of these steps and talk about what actually, the specifics of what happens at each step moving forward. So I mentioned we have different types of synapses. We have a chemical synapse, which is what we typically see. It's where there's a space and information is sent across that space from the presynaptic cell to the postsynaptic. So it involves the release of chemical messengers known as neurotransmitters. They carry information from the presynaptic, which is the sending neuron, to the postsynaptic, or the receiving neuron. Electrical synapses are rarer, but they're faster. There's a direct connection between pre and postsynaptic, and it takes place in the form of a channel called a gap junction, which allows current or ions to flow directly from one cell to another. Okay, so chemical, which is going to be more common. Almost every synapse that we have, by being human, is chemical. The characteristics, simplistically, are the first neuron is secreting a particular substance at the end of its own axon, specifically its axon terminal, called a neurotransmitter, which is then registered by a corresponding receptor on the dendrite, or even the axon of the second neuron, which produces some specific change. So going into specifically what happens, we see again on this picture on the left, we have our presynaptic cell and our postsynaptic cell. So there's an action potential, which we already talked about, that went through the presynaptic cell. It reaches the axon terminal, it depolarizes. Again, we're here, it's becoming more positive. The membrane, it opens voltage-gated sodium channels, they enter the cell, further depolarizing it. This leads to calcium channels to open. Calcium channels is our helper substance in getting the neurotransmitter across the synaptic cleft. So the calcium ions help with the release of whatever neurotransmitter it is, serotonin, dopamine, and it initiates a cascade that causes synaptic vessels containing the neurotransmitter to fuse with the presynaptic membrane. And you all are witnessing the joy of both me and my husband working from home, which means the door going off and my husband in the background. We've been like this for 2 years. So then there's a fusion of vesicles from the presynaptic membrane that causes the neurotransmitter to be released into the synaptic cleft, or that space in the middle. The neurotransmitter is going to diffuse across the synaptic cleft and bind to a receptor protein, which we're going to talk about on the postsynaptic membrane. This binding is really important. It causes ion channels to open, and this is where the action takes place. This is where we start the process of another action potential on the next cell. So it's this continuous loop. And Janet, I did see your question. As long as the moderators and Hawks is okay with it, I'm very happy to share my slides. And at the end, I can provide you with my email address individually too. We just want to do it that way. Okay, so neurotransmitters have different effects on the postsynaptic cell. So I told you it was going to get simple and then a little bit more complex. So When we talk about an action potential, I said it depends on if there's enough stimulus to reach a threshold of excitation. We're kind of going back in time here. Depolarization is when there's an explosion of electrical activity causing this depolarizing current where the cell becomes less negative. Hyperpolarization of the cell is when the cell becomes more negative. So there's less likely for an action potential to fire. All neurotransmitters are going to have a different type of effect. Some are excitatory that are more likely to lead to an action potential on the next cell, and some are inhibitory. So when we have what's the excitatory kind, it's what's called an excitatory postsynaptic potential, or an EPSP. It's the depolarization of the presynaptic membrane that makes the postsynaptic membrane more likely to fire an action potential. So it's more likely to keep up that process of action potentials, of signals being sent. The inhibitory is the opposite. We go back to hyperpolarization, so making the cell more negative and less likely for a postsynaptic neuron to fire an action potential on that next cell. So for example, we have acetylcholine, which is really important for learning and memory and for muscle movement. It's released at the synapse between a nerve and a muscle, which is called a neuromuscular junction, by a presynaptic neuron. It causes postsynaptic sodium channels to open. Sodium enters the postsynaptic cell and causes postsynaptic membrane to depolarize. This depolarization is called an EPSP, and it makes the postsynaptic membrane more likely to follow. On the other hand, if we have an inhibitory effect neurotransmitter like GABA, GABA would be the neurotransmitter that leads to like a more sedation calming effect. So people take, without getting too complex, people take something to help essentially boost the level of GABA at like the most elementary level to create an inhibitory effect to help with anxiety. So a benzodiazepine like a Xanax would help with that level of GABA. So GABA has this inhibitory effect. It's trying to slow things down. So a release of the neurotransmitter GABA at inhibitory synapse causes an IPSP, or inhibitory postsynaptic potential, or hyperpolarization. So when GABA is released from the presynaptic neuron, it binds and opens chloride channels, hyperpolarizes the membrane, and makes it less likely for that next cell to follow. Fire. I'm still drinking my morning coffee. Okay. So how do EPSPs and IPSPs interact? It's not as simple as just it leads to an EPSP, it leads to an IPSP. Basically, a postsynaptic neuron is going to add together, integrate all of the input that they have from the individual cell and decide whether it's going to fire an action potential based on the level of EPSP and IPSP. So spatial summation is the integration of postsynaptic potentials that occur in different locations, but at the same time. So you can have more cells and more information firing on a postsynaptic cell at the same time. So it occurs if there's multiple presynaptic cells together, releasing enough neurotransmitter, like acetylcholine, to exceed the threshold for the postsynaptic neuron. So for instance, let's suppose an excitatory synapse are made of two different dendrites from the same postsynaptic neuron. Neither synapse itself can produce an EPSP quite large enough to bring the membrane potential to the threshold. If there are multiple sub-threshold EPSPs occurring at the same time, it could sum or bring the membrane to a specific potential that it would be likely to fire an action potential. On the other hand, if you have an IPSP, an inhibitory, with one or two EPSPs, it might prevent the membrane from reaching a threshold of firing. So these are some examples of spatial summation. And then temporal summation. I see the questions. I'm happy to answer these at the end. I think that's typically what the protocol is. But yeah, I will answer them at the end. So temporal summation occurs when one presynaptic neuron releases a neurotransmitter many times over a period of time that exceeds a threshold on the postsynaptic neuron. So it's the same one acting over and over. So a key point to this is postsynaptic potentials aren't instantaneous. Instead, they last for a little bit before they dissipate. We're going to talk about how that dissipation takes place. But if you have a presynaptic neuron firing quickly twice in a row, like it has to go rest before firing again, it could lead to 2 EPSPs. The second EPSP may arrive before the first one is dissipated, bumping the membrane potential up. And this could be an example of temporal summation. Okay. So we don't want continuous action that's taking place on a postsynaptic neuron because that could flood it and lead to like toxicity. So an action takes place and then essentially the neurotransmitter has to go away. And we can use different classes of drugs to kind of alter how long there's action that takes place on a particular neuron. So it's not always the substance or the neurotransmitter itself, but it's often the time in which it's a combination of EPSPs, IPSPs, and the time in which it's taken away from the action of the postsynaptic neuron. So I said, before a neurotransmitter can be removed from the cleft. So after a neurotransmitter has been recognized by a postsynaptic receptor, it's released back in the synaptic cleft. Once in the synaptic cleft, that space in the middle, it must be quickly removed or chemically inactivated to prevent constant stimulation on a postsynaptic cell or excessive firing of an action potential. So say you have, excitatory neurotransmitter like acetylcholine that's continuously creating EPSPs, that could go away. It needs to do its job and go away. Kind of like me, or like give our talk and then you can go have lunch and then I'm coming back though. But do its job and then go and then do another job. So the neurotransmitter there's basically 3 things that can happen. One is diffusion. So a neurotransmitter will diffuse from the synapse into tissue and get absorbed by glial cells, which is the other type of neural cell. They're like the helper cells. Like if you're running a marathon, the marathon runners would be the neuronal or the neurons. And then the glial cells would be like the one giving them Gatorade and cheering them on and stuff. So it's those helper cells. Degradation is an enzyme can break down a neurotransmitter. So there's an enzyme, for example, called acetylcholine esterase within the synaptic gap that can break down acetylcholine, make it inactive. And then the breakdown products are recycled to be used again. Reuse, recycle. And then there's recycled or reuptake. So transporters are protein molecules embedded in the presynaptic membrane. And they're able to pump neurotransmitter molecules from the synaptic cleft back to the presynaptic cells. This process is called reuptake. It's really common for classic small molecule neurotransmitters, which we're going to talk about. But I think a good example of this is selective serotonin reuptake inhibitor, which is an SSRI. I know the question in the chat was about drugs. And so I think we've all heard of SSRIs. They help with depression. We don't totally understand why. We know that depression is associated with serotonin, and the fact that SSRIs, selective serotonin reuptake inhibitors, can work lends us evidence that there's disruption in the serotonin system. But selective serotonin reuptake inhibitors and also SNRIs, selective norepinephrine reuptake inhibitors, leave serotonin or norepinephrine in the synaptic cleft, taking action on the postsynaptic cell longer. So we're actually using a drug to change the action of the neurotransmitter. So this is where like the real world examples come in, and then that can help with depression symptomology. So quickly, which I don't think a lot of gen psych textbooks would go into this because they're not as common, but electrical synapses are fewer in number, but they're found in the nervous system and play an important and unique role. So instead of having that like synaptic space, the presynaptic cells and postsynaptic membranes are physically connected by channel proteins forming gap junctions. They allow currents to pass directly from one cell to the next, and they signal electrical synapses as opposed to, and their signal is virtually instantaneous. They're more reliable because they're less likely to be blocked, and they're important for synchronizing electrical activity of groups of neurons. So for example, electrical synapses in the thalamus, which is it's the relay station of the brain. It's like a main signal center to all different areas. For example, one part of it's really important for vision. So most of our visual information comes and goes to an area of the thalamus, is routed to the occipital lobe, which allows us to visualize things. But going back to the thalamus, that was just an off example. These electrical synapses in the thalamus are thought to regulate slow-wave sleep, and disruption can cause seizures. The benefit is they're fast, which can be important to help like an organism escape from a predator. So if there needs to be fast action like fight or flight, they can also be synchronized. In many cases, they carry current in both directions. So depolarization of a postsynaptic neuron can lead to depolarization of the presynaptic neurons. And then the downside is, unlike chemical synapses, electrical synapses can't turn an excitatory signal in one neuron to inhibitory in another. So it lacks some like versatility, flexibility, and capacity, but it is good for like an emergency system. All right, so we are gonna have some time. I'm gonna go over this quickly. But we're gonna talk a little bit about neurotransmitters and receptors. So I went too fast. So there's two different types of receptors on the postsynaptic cell that take in that information from the neurotransmitter. Ionotropic receptors are fast acting. They don't need any help. They're membrane spanning ion channels that directly open. They act quickly to depolarize the neuron and pass on an action potential or hyperpolarize if the neuron inhibits. The other type, metabotropic, are not ion channels in themselves. They need a little bit of help. It depends on one or more metabolic steps, hence why it has the name. The receptor complex includes what's called a transmembrane receptor protein, or a G protein. It's the first messenger that binds to receptor protein and helps initiate the process of going from one area to another. I don't spend too much time on this because we're running out of it. So this is just a longer version of that, but quickly let's talk a little bit about neurotransmitters. So neurotransmitters are the body's chemical messengers. They're what's being passed on from the presynaptic to the postsynaptic neuron. They're molecules used by the nervous system to transmit messages from neurons, between neuron to neuron or neuron to muscle to like tell our arm to move. The signaling at the synapse brings about a response that depends on both the neurotransmitter from the presynaptic cell and the receptor, how it's received from the postsynaptic cell. And a single neurotransmitter may have more than a dozen different types of receptors. So I want to talk about a few different common types of receptors, or neurotransmitters. There are a lot. There are many different kinds of neurotransmitters, with new ones being discovered all the time. And over the years, the very idea of what makes something a neurotransmitter has changed. So by definition, it's been expanded. We have what's called traditional, and then we have non-traditional or unconventional neurotransmitters. So some of our conventional neurotransmitters share basic functions. These would be considered like serotonin or dopamine. They share these common features. They're stored in synaptic vesicles. They get released when calcium enters the axon terminal in response to an action potential, and it can act by binding to receptors on the membrane in the postsynaptic cell. Small molecule neurotransmitters that we're going to talk about, which are common neurotransmitters and include acetylcholine, amino acids, and biogenic amines. So acetylcholine is a really common neurotransmitter in both invertebrates and vertebrates. It's vital for functions that involve muscle stimulation, memory formation, and learning. Acetylcholinesterase, which is can help. It's a building block to acetylcholine. It can be used to help with memory problems. And then there's two different types of acetylcholine receptors. We have both the ionotropic, the ligand gated, or the metabotropic. A number of toxins can disrupt neurotransmitter or neurotransmission of acetylcholine, like nerve gas, sarin, or bacterial toxin from botulism, which can impair movement. Amino acids, some examples include glutamate, which is the most common brain neurotransmitter. It regulates development and creation of new nerve pathways involved in learning and memory. GABA, which inhibits neuron firing. So high levels can help improve focus, whereas low levels can cause anxiety. So we try to increase the levels of GABA, which I know is one of the questions in the chat. And it also can contribute to motor control and vision. Biogenic amines. They are active in both the central nervous system, which is the brain and spinal cord, and the periphery. These are ones you're typically going to talk about. I know I'm out of time, but this is our last slide. So this is typically what would come up in a neuroscience class, as these are typically part of like our major drug classes for mental disorders. So neurotransmitter is predominantly a neurotransmitter. It's occasionally released as a hormone, contracts blood vessels and increases blood flow, can help with improved attention and speed in which a responsive action occurs. Epinephrine is primarily a hormone released by the adrenal gland. Some release it as a neurotransmitter. It increases heart rate, blood flow, leading to physical boost, heightened awareness. So, you have that like adrenaline boost. It's epinephrine boost. And it's produced during stressful or exciting situations. Dopamine is responsible for feelings of pleasure. So I say, when I have a cookie, I have a dopamine rush. And I want more cookies because I want more of that dopamine. It's Girl Scout cookie season, so it's really dangerous around here. But it's also involved in movement and motivation. Abnormal secretion can lead to issues, and it's common in Parkinson's disease and other movement disorders. People tend to repeat behaviors that lead to dopamine release. So like it can lead to addiction, like eating too many cookies, cigarettes, opioids. Serotonin contributes to feelings of well-being and happiness. We know there's an association with major depressive disorder, and it's affected by a lot of things that we can, this is ending on a positive note, that we can do to naturally boost that action of serotonin at our receptor level. So like exercise, light exposure, and it also plays a role in sleep cycle and regulation. So I always get rushed at the end of this talk. I should cut it, but if you have any questions or you want a copy of the PowerPoint, I'm happy to send it. My email address is there. I'm Lori Slifka, and then at kaiseruniversity.edu, and I'm happy to provide you with any materials that I have. It's totally up for collaboration and sharing. It's one of my main roles is faculty development. So if I can help in any way, I'm happy to. So I hope you learned something what got rushed at the end. I'm happy to take questions.
Yes, thank you so much, Lori. It looks like we do have a couple of questions in the chat. I'm not sure if I'm going to pronounce this right, but Lynn asked, is GABA found in Suboxone, opioid inhibitor? At least that is my understanding.
So I saw that question before, and I haven't studied psychopharmacology since grad school. So my answer to that is I am 90% sure that GABA is found in Suboxone, and it is one of the main actions of Suboxone. But I'm not 100% sure because it's been so long since I've done anything with opioids. You know what I mean? Mm-hmm. Not in that sense, but I'm happy to look at it. I'm happy to talk about it further, look it up with you if you email me. So it's in terms of the anonymous attendee. So serotonin is what's considered a conventional neurotransmitter, but it's in one of those classes. So there's conventional neurotransmitters that fall into the classes that I named, including biogenic amines.
Gotcha. Any other questions? Feel free to enter those into the Q&A and we can address any of those. But thank you so much, Lori, for all the information that you shared with us today. We will, our next session is the lunch keynote speaker. It's Arjun Buxi. So you can View the chat for any of the session meeting room links or access to the conference website for a complete list of concurrent sessions and their descriptions. While accessing the conference website, don't forget to swing by the exhibit hall to say hello to the Hawks team, view a quick 5-minute demonstration, and be entered to win the hourly giveaway, your choice of 6-month subscription to Audible, 1-year subscription to Disney Plus, or $100 Amazon gift card. But it looks like we don't have any other questions at this time. Thank you so much again, Lori, and we hope to see everyone in your next session.
Yep, thank you so much, Sarah. And